My name's Trey Eidecker. I'm the contact PI for the functional genomics DGP known as Cell Maps for AI. I'm going to tag team this with our program manager, Jillian Parker, who will take over about halfway through the presentation. And what we really think about in a sentence or two, if you boil it down in Cell Maps for AI, is how do we implement a precision medicine AI system that's fully explainable? So let me explain what I mean by explainable precision medicine. So I think all of us, and I think that includes probably some of the groups up here as well, are going for one day in the near future, we think, systems that live in phones and on computers where you can speak to them about patients and given the patient data that you've collected, and in our case, we're the functional genomics project, and so we think a lot about genetic data collected for patients. How does that get translated into predicted outcomes and diagnoses? And so, you'd like to have a system that looks like this. Dear AI, my patient has the following data frame. Here again, I've included the genome sequence in that data frame, of course. What treatment is best? And if you're in a drug company, maybe the better question is, should I look at new targets, or are the existing drugs sufficient for this patient? So that's precision medicine, in terms of an overall vision. But the key follow-up explainability question would go something like, why do you think that? Just like a physician or a scientist or anyone is called to task to explain their views, these systems cannot be wizards of Oz behind green curtains. They need to be held to task to discuss and explain their points of view. And so often in biomedicine, those explanations are a molecular explanation of that underlying condition, or at least a cellular explanation of that underlying condition. So for instance, in the cancer clinic, maybe the recommendation is give an AKT inhibitor. Why do I think that? Well, this patient doesn't have a mutation in the target AKT, but they do have a mutation just upstream of AKT, making me think, and especially because I've seen patients like that before, that they'll be responsive. So that would be an example of the kind of explanations we're going for here. And so what that means here is we need a middle layer in this genotype-phenotype equation in between. So rather than sort of take the raw molecular genome sequence and clinical history and go directly to predicted outcomes and diagnoses, we really want to go through one, and it turns out likely multiple tiers of intervening information. If you think about this as a physics problem, genome mutations and variants exist at the level of nanometers. Patient phenotypes exist at the level of meters, right? Order of magnitude. So we're talking about bridging 9 orders of magnitude without any intervening data. Really? Okay, that's really the prospect and the opportunity here for our Bridge2AI project is, what data do we need? Thank you. What data do we need to fill in the gaps here between patient genomic information and patient outcomes? And so the data we're right in the middle of generating right now is to take relevant cell line examples of human cells and generate those molecular maps onto which you can project patient molecular data. And so our models so far have been a triple negative breast cancer cell line in the naive state and after exposure to 2 prominent cancer therapies. And if you think about the mother of all human cells, the human-induced pluripotent stem cell. And in fact, we're now talking with some of the other Bridge2AI projects about generating iPSC cells from some of their donors, which is an exciting potential moving forward. Here we've started with the most standard and used iPSC cell currently in the public domain. And once again, we can look in the naive, undifferentiated state, as well as, in this case, 2 representative differentiated programs, neurons and cardiac myocytes. Those are the cell models. And then each of those, if you think about it, 3 and 3, so 6 different model systems, if you look at those 2 triangles at the left, are now interrogated via 3 different omics data layers. First is confocal protein imaging to light proteins up in those different cell populations and to try to do so systematically over hundreds, if not thousands of proteins. The second is protein-protein interaction complexes measured by mass spectrometry, again, in those 6 models. And the third is so-called perturb-seq, where you knock out each gene in turn and measure its effect on transcription. So Jillian, please take it from here. Oh, okay. Just quickly, and then we integrate those three data streams into maps of human cells. So this would be a multimodal map integrating, in this case, two, and the next version will have all three data streams integrated. So thus far, we can resolve in the cancer cell type about 275 assemblies, or exactly 275 assemblies. And as you see here, these range from large assemblies up to organelles, all the way down at the bottom of the page here to small protein complexes that live in each of those subcellular compartments.
Okay, so I'm going to talk a little bit more in detail about our current data generation progress, data releases that we have planned upcoming, as well as some additional ways to engage with our data and tools that we are producing as part of this project. So in this graphic, what you'll see is the current data progress about 2.5 years into the project. So what I want to mention here is that when we originally proposed this project, our goal was going to be to collect data from all of these three streams on a core set of 200 proteins that would be shared between all of the platforms so that we had a cohesive data set that is multimodal. With the economies of scale that we've been able to achieve so far, we have blown way past that 200 protein goal. And we're actually collecting, in some cases, whole proteome interactomes and whole genome perturb-seq data as well. So you'll see the updated numbers for our current data collection plan on the slide. And the colors of the boxes indicate our progress towards total data collection. So as you can see, the breast cancer cell lines are nearly complete in terms of data collection across all of the modalities. And we're well on our way towards collecting data in the iPSCs, both in the parental state, which we've done quite a deep dive in, as well as some of the differentiated states that Trey mentioned. In addition, I do want to point out that because of the economies of scale that we've been able to achieve, we also have now included some intermediate states to differentiation. So for example, going from the parental to the neural, there's an intermediate state called NPCs, and we've been able to collect data on those stable NPC cell lines as well. And then just in terms of data releases, we did a big bulk data release of raw data for all of the modalities this past March. If you scan that QR code, it will take you to Dataverse where the data live, packaged nicely with the metadata from Tim Clark's team using their Fairscape client, which you'll hear more about later today. And then going forward, we're planning on doing quarterly releases of at least some data types as the data become collected and QC'd and are ready for the limelight. And just to point out some additional numbers, it might not look like, if you just look at the number of proteins or genes that we're collecting, that there's that much data. But for example, in the images that we released in March, the data set actually captures over 11,000 single images, capturing over 100,000 cells, very large data set. For the SecMS data that we released, it's actually more than 5,000 proteins per replicate, multiple replicates released. And then for the whole genome perturb-seq data in the parental iPSCs, the raw data is capturing whole genome level perturbation. So it's over 11,000 genes. And it's actually such deep coverage that it's equivalent to 30x genome sequencing of 450 individual human genomes. So it's quite impressive and quite large data in terms of size. So just really quickly, some additional ways to engage with our data and tools. Obviously, I mentioned Dataverse. You'll hear more about that later. We also leverage domain-specific repositories. So if you're used to working with perturb-seq data, you'll probably look on SRA. You will find our data there. And then in addition to interacting with the data, you can interact with some of our tools via our website. They're all open source. And some of our additional platforms, such as Cytoscape, we're about to launch a web version, which will allow you to look at both the hierarchy tree and the circle packing view in the same window, and see all of the underlying data supporting those protein assemblies. For now, we have this collection of our music maps that you can view and index at that link there. Also working with CFDE and Uniprot to integrate all of our data and tools. And then just to wrap up really quickly, we hope you join us at our Code Fest this afternoon. that will be led by our skills and workforce development team. We are very excited. We just launched our first public Kaggle challenge online last week. This one is focused on community detection using some of our recently released data. And then check at our website for additional updates. We are planning a ton of virtual and in-person challenges using our data with some of these focus areas that you see listed here. Of course, we're always interested in hearing about your use cases. If you can think of something that you would love to do with our data that's new and interesting, please feel free to reach out. My email is there. You can email me directly, or you can visit our website and get in contact with us there. Thank you.